So when we talk about blue-green algae, even though they're called algae, technically blue-green algae, which are also called cyanobacteria, are a type of bacteria very different than most algae, which are actually eukaryotes. And if you're familiar with the term eukaryote or prokaryote, a eukaryotic cell is we're eukaryotes, animals, plants, fungi, various protists. We have all sorts of organelles. We have a very complicated looking cell compared to a prokaryote, which are bacteria, which do not have all the organelles you see in a eukaryotic cell. So if you look at eukaryotes can include bulldog puppies, my daughter Anna, or a bloom of euglena. But look at this. This is a bloom of euglena, yet this is a bloom of Anabina, which is a blue-green algae. So just looking at the scum, they look the same, but my daughter and the bulldog are actually more closely related to the euglena than the euglena is to Anabina. And this is really important from a management standpoint, because if we're trying to manage or correct this problem, the recommendations we would make for this type of bloom are very different than a blue-green algal bloom. The other thing is this bloom will produce cyanotoxins, where this one will not. So again, it's critical we know what these organisms are and look like. So again, the term algae is more of it's not a taxonomic term as much as it's an ecological term. It's representing the group of microorganisms that photosynthesize. So it does include blue-green algae, which are also known as cyanobacteria. Now, algae are very important in aquatic ecosystems. They're the base of the food web in many aquatic systems. They contribute oxygen, they sequester carbon. They are being used for biofuel, and they have some pharmaceutical values. However, some algae are problematic. And when we talk about algae from more of a morphological basis, we're talking about three main types of algae. There's what we call phytoplankton, which are the free-floating algae. There's the filamentous mat algae, the benthic algae. A lot of times I'll describe it as that green cotton candy you see in water. And there are a few green algae that are called macroalgae that actually look like plants, but stoneworts, they are really algae. So the ones we'll be focusing on primarily are the phytoplankton. There are some blue-greens that are also benthic, but we'll be focusing mostly on the phytoplankton in the large, more riverine systems. So when we talk about algae in terms of having a negative impact, in terms of producing some type of toxin, we call them harmful algae blooms. And in marine situations, you can have organisms like diatoms or dinoflagellates that can produce some nasty toxins. But what we're really going to focus on are the blue-green algae, which really plague the freshwater environments. So obviously they have a direct impact not only on recreation, but they also have a direct impact on drinking water supplies as well. So again, when you look at algae, there are all kinds of groups. There are the green algae, chrysophytes and diatoms and dinoflagellates, which can make the water look brown or can make it look red. Euglenoids, which can make it green or red. But we're really going to be talking quite a bit about the blue-green algae. Again, the ones that can cause most of the problems in freshwater environments. So when we talk about blue-green algae or cyanobacteria, again, they're photosynthetic bacteria. They're very old organisms. They're probably the oldest known organisms in fossil record history. They are the dominant nuisance group in freshwater systems. And they're responsible for the majority of the surface scums you see that affect recreational systems, as well as potable water supplies. They can produce cyanotoxins and taste and odor compounds, and many are not grazed by the zooplankton, the little microanimals that will actually eat a lot of the algae in the water column. So since blue-green algae have been around for so long, they have some pretty unique adaptations to surviving in freshwater environments. For example, they can photosynthesize a wide variety of light intensities. And that's important because if you think of a river system, if there hasn't been rain for a while, the water may clear up, but if you have a big storm and it's bringing in a lot of suspended material, the light intensity goes down in that river. Well, cyanobacteria can photosynthesize in a variety of light intensities. Also, many of them can fix atmospheric nitrogen with little specialized cells called heterocystses. They can regulate their position in the water column with gas vacuoles, and they generate colonies or these cyanotoxins, which make them unpalatable to the zooplankton. Now again, some of these I'll get into a little more detail as we go through. So one thing they do is they can produce these specialized cells called akinetes. And akinetes are resting cells. So when the temperature starts to decline and there's not as much light, or in a lake situation it freezes over, these cells, these akinete cells, will sink to the bottom. And they're very resistant to environmental conditions. They have thick cell walls, and then in a lake or even a riverine system, when those akinetes are washed back up or transported back up to the surface, there's more light, they'll essentially hatch and you'll have more blue-green algae. So they survive these very harsh environmental conditions, and when a lake mixes or you have some riverine upwelling, you can have the germination of new blue-green algae. This is what an akinete will typically look like. So this is a filament of Anabena. These are the vegetative cells. The akinete, it's a larger, thicker cell wall. That's that surviving spore that will sink to the bottom. I mentioned the heterocystses, again, thicker cell walls. A heterocyst, though, inside the cell there's no oxygen, and it provides the biochemistry for the blue-green algae to actually fix atmospheric nitrogen. This is very important for a lot of blue-green algae, because that means what really dictates the growth of blue-green algae is phosphorus and not necessarily nitrogen. When you have elevated nitrogen going into more marine or estuarine areas, that can really have a negative impact on terms of developing more algae blooms. But in freshwater systems, most of the problems associated with freshwater tends to be blue-green algae, which means phosphorus. So in a lot of instances, in a lot of lakes, reservoirs, or rivers, we tend to focus a lot more on the phosphorus than the nitrogen. If you can if you can pull nitrogen from the atmosphere with these heterocystses, you don't care how much nitrogen is in the water column. But to do that, you need a lot of phosphorus. So that's why when you see an increase of phosphorus, not only do you see more algae, you tend to see more blue-green algae. So again, this is how they do it, through these specialized cells called heterocystses. Therefore, a lot of the blue-greens, not all, but many, are not dependent on external sources of nitrogen, such as nitrate or ammonia. And this is what a heterocyst looks like. It's a clear cell. See all the vegetated cells? That's where it's fixing atmospheric nitrogen, producing nitrogen for the rest of the filament. So studies have also shown that the frequency, the number of heterocystses on that chain, is dependent on the availability of the dissolved inorganic nitrogen. And in terms of fixing nitrogen, like I mentioned, it's a very energy-demanding process. So you need more phosphorus to really do that. Another adaptation they have are gas vesicles, which in riverine systems initially may not seem like a good adaptation, but it can be successfully employed, particularly in low-flow environments. But essentially, blue-greens can produce gas vesicles, which are basically clusters into these gas vacuoles. It's the way the algae can move up and down the water column. So the reason why an algal will want to move up and down the water column, this is just a sample of an algae during a bloom. You can see most of the cells are up toward the top. If you're competing with other algae for light, you can float to the surface and harvest all the light you want. And then once you have all the light you need, a lot of times the more nutrient-rich waters are down deeper, so then you can collapse those gas vesicles and float on down and take all the nutrients you want. So it's really an adaptation to harvest both light and nutrients.
Is that limited to cyanobacteria?
It really is, yeah. So most other algae have other adaptations to try to survive in the water column. Many green algae have flagella, so they attempt to keep swimming as much as possible. Many cells are very small, or they'll have unique shapes to sort of resist the gravity, pushing the cell down. But it is unique for blue-greens that they actually have this ability, and they can move up and down the water column over the course of a day. So a lot of our drinking water facilities know at 3 o'clock in the afternoon, I know where my intake is, I'm going to have a problem with blue-green algae if I don't do something about it. Because the cells concentrate, they may concentrate at the surface or they may move toward the bottom. And it's all whether or not they're producing these gas vesicles. So on a daily basis, a lot of times over the course of the evening hours, when you're going into morning, they'll float right to the surface. They'll harvest all the light they want, and then as you move into the afternoon, they'll start moving down. So a lot of drinking water supplies know where my intake is. I know I may have some problems with blue-green algae moving up and down the water column. And this is just showing the actual mechanism, how it operates is, it's not like they're thinking, I need to move up, I need to move down. When they're up toward the surface, as they photosynthesize, they're producing carbohydrates, which makes more pressure in the cell, so it makes the gas vesicles collapse. Cells get heavier and they sink. Once they're down here and they're using up those carbohydrates, there's more room in the cell to create those vesicles. So they start making them, they're harvesting all the nutrients, and then they float right to the surface. So it's this biochemical response to the environmental conditions. Here are some of the more common ones. I'll show you some microphotographs of what they look like. Some of the more common ones, Anabena, which there's some taxonomic changes going on with Anabena, but we still very frequently call Anabena. We'll commonly call it Annie, and Phannozomenon, Fanny, Microcystis, Mike, and then Celospherium, Cilia. But these are some of the more common nuisance blue-greens. Anabena, when you look at it, it will be these bundles of filaments. They'll be very long and thin. The heterocysts will be long, thin, and narrow. And if you're looking at a water body with a bloom of this, it will look like someone threw a bunch of grass clippings in the water, because you can imagine this looks like one big grass clipping in the water. And this is, a more close-up view of the cells. They tend to be either rectangular or square, and they'll have these longer akinetes or longer heterocysts. Anabena, which is a very common one, it sort of looks like beads on a chain. You can see the heterocysts, very common in terms of producing cyanotoxins. Again, I've showed this before. There's a heterocyst. Cylindrospermopsis, again, it's more of a narrow and thin cell, and it have heterocysts very frequently on the ends. I put this one out. This one's sort of rare in our neck of the woods, but there's a cyanotoxin named after it that EPA is concerned about, Cylindrospermopsis. So I do put this up there, even though it's not as common as some of the others in this part of the United States. Gliotrichia, which is more of a colonial one. When you see this one in the water column, they'll look like little beads, little spheres in the water. Microcystis, which is one of them that will not produce heterocysts. So it's more dependent on nitrogen. So very frequently, what we'll see in a lot of systems is the ones that can fix the nitrogen bloom first. And then once that bloom starts to senesce, a lot of times when you get into the late summer, early fall, mid-fall, the microcystis will very frequently take over, because as that other bloom is senescing and dying off, it's releasing nitrogen and phosphorus into the water, and microcystis will take advantage of it. This one will look very gloppy, very globular in the water. Celospherium, which again is one that does not produce, it does not fix nitrogen, but the Celospherium, as far as I know, does not produce cyanotoxins, but it's starting to be separated from a genus that is known to produce cyanotoxins. So this one is a genus that's sort of in the process of literally being separated into two different genera. And then Oscillatoria is another common one. This one will very frequently be more of a benthic algae, so it will form more like mats that will hang out at the bottom, and very frequently these mats will detach from the bottom, float to the top, and then once they hit the surface, Oscillatoria and some of the other filaments like this that look like a stack of coins, they don't do very well when there's a high intensity of light, so they'll start to decompose and die. You'll get nasty taste and odor compounds, you'll get unpleasant colors and appearances, and a lot of times it's not the bloom, but it's the senescence of the bloom that looks really bad. So some additional information on these blue-green algae. Most prefer higher water temperatures and tend to bloom during the summer months. Most tend to thrive when you have phosphorus-enriched waters. So if you're in a lake system, if your concentration is like 0.03 milligrams per liter or higher, that's when you have the chance of producing these blooms. They do prefer a still water habitat. And you can figure that out, especially the ones that have the gas vacuoles. If it's a nice still water habitat, it allows the blue-greens to move up and down the water column. If it's a well-mixed system, it's more difficult for them to create those blooms. So a lot of times in lake and reservoir systems, we install de-strat systems to keep the system well-mixed. So it can help to control phosphorus coming up from the sediments, but very frequently what all that's doing is it's eliminating that still water habitat, so it makes it difficult for the blue-greens to make those concentrated blooms that can move up and down the water column. Most do not do well in acidic waters. So when we look at lakes, say in the Pinelands, or lakes up in the Poconos that tend to have are a little more acidic, a little low alkalinity, we tend to see other algae that can be nuisance, some green algae, filamentous green algae, or some of the chrysophytes and diatoms can be problematic. But in most riverine systems that have low flow environments, it's the blue-greens that can really easily take over. And they are generally more susceptible to copper sulfate. However, there have been studies that, depending heavily on copper can increase the tolerance of copper by these organisms year after year. If all you're doing is just hitting it with copper, you actually make it less cost effective because they have an increased tolerance for that algicide. So the three conditions that almost always will result in a blue-green algae bloom in a freshwater system is high water temperatures, still water, and elevated phosphorus concentrations. So when you think of that, you think of a riverine system, you're like, well, yeah, we can have high seasonal temperatures, and we can certainly have high phosphorus concentrations, but we usually don't have still water conditions in a reservoir system. So can blue-greens bloom in rivers? And yes, they can. They can thrive in these environments. And that you would think it was very rare, but it's been well documented that they can bloom quite frequently. And a lot of it has to do with more low-flow conditions. So in a lake perspective, where these blooms may be very routine and periodic, in a riverine system, the blooms are more dependent on the hydrologic conditions of the year. So if you have a low flow condition where the riverine systems are slowing down in terms of the amount of flow, that slow meandering river can really result in some pretty nasty nuisance cyanotoxin blooms. So this is something that, for this presentation I really wanted to discuss, how would you deal with these blooms and why are we concerned about them? Now, if you're a recreational person, if you're a boater out there, you certainly don't want to be out there. It's an unpleasant condition. You have unpleasant odors and appearances. If you're a drinking water supplier, though, you're concerned about how all these cells are going to affect your filter runs, how it's going to increase cost, in terms of treating that water to make it drinkable. But something that has really come up over the last, I would say, 5 to 10 years has been the concern over cyanotoxins. So that's really what I'm going to focus on for most of this. Now to do that, I really want to talk a little bit about the river continuum concept. So I'm sure everyone is familiar with this. You have your headwater environments way up high, and actually if you're at the keynote presentation, they did focus on this quite a bit, that those headwater environments, you have very clear, clean water if it's in a forested area. In those situations, you tend to have a canopy of trees. So the source of carbon for those streams tends to be the leaf litter. It falls down into the stream. And then you have these shredders, these macroinvertebrates, all the little aquatic bugs tear that leaf litter up. They use it as a source of carbon, and as tearing it up, they make more surface area for bacteria and fungi to break all that down. So really the base of the food web up in these headwaters tends to be the leaf litter. Leaf litter falls down, and that really keeps this process going. As you move from a high order to a low order to a high order stream, as you're going into these wider stream areas, you have more sunlight hitting the bottom of the stream. As you have more sunlight hitting the bottom, that's when you start to have plants or algae growing on the stream bed. So you'll start to see grazers. So here's where we'll start to see more of the snails, water pennies, some of the caddisfly larvae that actually are feeding on the algae or feeding over the aquatic plants or the stones. You'll still have the shredders and the collectors, but you're seeing this transition from the base of the food web being the leaf litter, going over here you're moving more to plants and to algae. And then when you go down a little further down the Delaware River, if you've ever been tubing and there hasn't been a storm for a few days or weeks, you can see some nice clear water, and you'll see these beds of Alidea, some submerged aquatic plants. But then as you go further down, the water gets deeper, there's more nutrient input, By the time you get down here, it's almost all phytoplankton. So now we have an algae-dominated system. Tends to be more turbid. There's no light getting to the bottom. So it allows the phytoplankton to grow and shade out any submerged aquatic plant growth. So you have this transition from leaf litter to a combination of algae and plants, maybe some more plants, but then you'll really get the phytoplankton. And this is, down in the larger stretches where not all, but a lot of your intakes are. So they're very concerned about what's going on in these riverine systems. And like I mentioned, if you have a very dry year and those flows tend to be very low, they slow down, it can easily shift that phytoplankton community from like diatoms and greens to the nasty blue-green algae. So this was, I think, 2006. There were a number of articles. If you remember this was late summer of 2016, and 2016 was a very dry year, and we had some record blooms in a lot of the lakes and reservoirs that we study that occurred in September, October, and even in early November. And so everyone was very cognizant in 2016 of blooms. Well, this occurred along the Schuylkill, and there was concern, was this a toxic blue-green algae bloom? It turns out, no, this was a bloom of duckweed. So it's a tiny, small, floating plant. Does not produce cyanotoxins, but it was a big scare. But it does emphasize that, 2016 was a very dry year. It had very low flows. And what the scientists who investigated this hypothesized is there was a small storm that washed some duckweed in an upper area. It came down along the Schuylkill, and the flow was so low it allowed the duckweed to bloom. Well, if this can happen with duckweed, this could certainly absolutely happen with blue-green algae blooms. So yes, this was a bloom of duckweed, but what we're seeing is that as we move into summer, fall, we're seeing more of these blooms occurring later in the fall. This year was the third warmest September. If you look at NASA's long-term climate change data, the first and second warmest Septembers were 2016, 2014, and then the fourth was 2015. So what we're seeing is, especially what we see, is that these blooms where 10, 15 years ago, they may end once you hit into mid-September, everything would crash. Now we're seeing these blooms going well into October and even early November. We had a reservoir up in New York we worked on that they had cyanotoxin blooms going into like the second week of November of 2016. So this is something that is an issue of concern. We're seeing this growing season get longer, and it's allowing these blue-green algae to bloom for longer periods of time. So again, in this case, what the scientists who investigated this hypothesized that more than likely some small isolated storms washed the duckweed into the river, and with the seasonally low flow, it just allowed the plant to bloom. But like I said, this is something that is of concern relative to blue-green algae and the potential production of cyanotoxins. So just to give you a little information on some of the potable water intakes for the Schuylkill River watershed, there are three major ones. There's actually four, and I'll show you a graph in just a second. There's the Philadelphia Water Department, Aqua Pennsylvania, and Pennsylvania American Water Company. Combined, these three serve about 1.5 million people in the Philadelphia area. And there certainly are other water intakes in the watershed. So the three major ones, 20% for Aqua America, and then Philadelphia 60. But I was surprised to see that PA American Water Company was about the same as Reading Area Water Authority. So I would say there was more like four major sources, and where the intakes are located. So they're, and again, these are surface water intakes. This isn't groundwater, but they're found throughout the entire watershed. So you can imagine that again, thinking of that keynote presentation this morning, any impacts on land use that reduces the quality of the water going into receiving waterways, the streams, the headwater areas. Eventually, when it all gets down to these lower reaches, it definitely has a negative impact by putting more nutrients into the water column, negatively affecting the drinking water and also resulting in increased costs. Again, I found that very interesting with the keynote speech. He mentioned that a 10% reduction in land cover results in an 8 to 20% increase in the treatment of that water. Again, any algae in the water column to a water purveyor is problematic because more algae means you have to filter more and you have to treat more, but they're very concerned about the cyanotoxins. And I definitely want to emphasize that it's not as simple as it sounds. Just because you have a bloom doesn't necessarily mean it's a harmful algae bloom or they're producing cyanotoxins. So this is a situation where you really have to have something in place where you can get an idea or a sense of, number one, what type of algae you have, and number two, a way of assessing whether or not you have cyanotoxins. I also want to emphasize cyanotoxins are not taste and odor compounds. Cyanotoxins are colorless, they're odorless, they're tasteless. You don't know if they're in there. You actually have to measure them. This is something that we have to constantly remind anyone we talk to, even our water purveyor customers, because there are compounds called geosmin and MIB that do produce, and they're produced by cyanobacteria and some actinobacteria. But these compounds are taste and odor compounds, but they're not health problems. They're similar to the compounds when you smell rotting fruit as fruit begins to rot. So they'll have like an earthy or a fishy odor. It's an off odor, off taste. They are not health problems. And so just because you have an off odor or taste does not mean you have cyanotoxins. And that's something we've had to work quite a bit with water purveyors and their customers, emphasizing that if you really want to know if you have cyanotoxins, you really do need to test for it. Just because you have taste and odor compounds doesn't mean you have cyanotoxins. And just because you have cyanotoxins doesn't mean you have taste and odor compounds. Unfortunately, there's no simple relationship between the two. So what are cyanotoxins? I've been talking about them. They're a group of chemical substances produced by blue-green algae. There are three main groups. There are neurotoxins, hepatotoxins, and dermotoxins. So neurotoxins affect your nervous system. Hepatotoxins affect your liver. Dermatoxins are more of an allergic reaction. They are well documented to have impacts on humans, livestock, and pets. And I was actually at the Pennsylvania Lake Management Society meeting, and there was a scientist there who was using Sea Grant funding, and she actually provided some information that pets tend to be more susceptible to cyanotoxins than humans, just on a concentration basis, which I found interesting. The concentration of, say, microcystin, which is one of the most common cyanotoxins that affect pets, is actually below the limit of detection of a lot of the methodology. They tend to be very sensitive. So in terms of dog parks, that's something we're really cognizant of, because if you're playing Frisbee with your dog, it's a hot summer day at a park, that dog, if it's hot, it's August, it's going to go to the water. It doesn't matter what color that water is, it's going to drink that water up. So there are very frequently situations where pets, or in an agricultural setting, livestock, drink a lot of these algal cells with cyanotoxins, and they get sick or even die. So there are concerns with cyanotoxins. There's drinking, bathing, contact recreational activities. There was a presentation up at the Aquatic Plant Management Society in January. There were a couple papers that showed that the contact recreational activity isn't just dermal, but if, say, you're jet skiing, water skiing, boating, swimming, a lot of the water tends to go into the air. That can go aerosol. It can go into your nose, into your mouth. If you have a fountain aerator, and the wind's blowing, and you're at a certain section along there, that fountain aerator may be putting these cyanotoxins into the atmosphere, so something to keep in mind. So again, contact recreational tends to be more associated with skin rashes, but we are concerned with any other issues associated with neurotoxins or hepatotoxins. And there is some increased attention over the possible links. The studies are literally going on right now on any sort of links between cyanotoxins and some of these neurodegenerative diseases. But again, there's nothing definitive. The one thing I can say is they're actually doing studies on this right now. So the cyanotoxins are released into the environment by both living and dead cyanobacteria. But from a water quality perspective, for a water purveyor, if you can keep those cells intact as much as possible, that really helps to minimize the issues. So if you can keep the cells intact, maybe filter them out, settle them out, a lot of times you can remove the bulk of the cyanotoxin out of the water. And I'll show you an example of some studies we did that demonstrates the importance of that. They are relatively stable and they decompose slowly. So once you have them in there, if they're in the cells, you want to try to keep them in the cells as long as possible. But if you know there's a problem, you need to be pretty proactive fairly quickly. I'm a limnologist. I'm an algae person. In the company I work with, they call me Dr. Scum because I work a lot with algae. And we've been talking about cyanotoxins for years now, but what really pushed this issue to the forefront was in August of 2014. You're mentioning, Lake Erie, a massive cyanobacterial bloom on the western end of Lake Erie, where, the cyanotoxin microcystin was not only found in the raw water, but it was actually found in the municipal water. So 500,000 people were warned not to drink the water. They couldn't even boil it to really break it down. So this was a major issue. And this is Lake Erie. This is showing it in August of 2014. And you can see that off-color green, that's that bloom affecting 500,000 people. And that's really what sparked this concern. So you can imagine changes in the watershed in terms of bringing in more nutrients, long-term climate change, and this dependency of this water is really sort of this intersect of how cyanotoxins can potentially impact people. So currently DEP, Pennsylvania DEP, does not have a standard. World Health Organization had a provisional drinking water standard of 1 microgram per liter or 1 part per billion of microcystin LR, which is one type of microcystin. New York DEC actually does have its own criteria for microcystin LR. They have the same for the World Health Organization of drinking water. They have a contact warning of if it's greater than 4, and then a beach closure if it's greater than 20. But in May of 2015, US EPA developed health advisories for 2 specific cyanotoxins in drinking water supplies. In addition to that, for those 2 types of cyanotoxins I'll talk about, they also released health criteria, ambient water quality criteria for recreation or swimming. So, and that happened in November of 2016. Currently, there are a total of about 10 cyanotoxins or group of cyanotoxins on EPA's unregulated contaminant monitoring rule. And this monitoring is going to occur between this year and 2020. So any drinking water facility that that provides drinking water for either 100,000 or more people, they have to be involved in this program. So this is something that's going on for the next 2 years, that EPA is collecting all of this data. But meanwhile, they do have some health advisories. And I can tell you, for the drinking water companies we've been working with, they're taking these health advisories very seriously. Even though they're draft health advisories, they are really focusing on them, because the last thing they want is someone accusing them of letting these cyanotoxins go through the systems. So there's a 10 drinking water day health advisory. The 2 specific cyanotoxins that are listed are microcystins, so it's all microcystins, not just LR, and then this cylindrospermopsin. And when EPA first released this, they had 2 criteria. They had, for microcystins, children younger than 6.3, and then for anyone older than 6.7, for anyone older it was 1.6. Then they also had it for the cylindrospermopsin. But these 2, a lot of the water purveyors didn't like that. They said, why are you giving us 2 criteria? We're not going to tell someone your 6-year-old can't drink the water, but we're going to say your 7-year-old can drink it. So all of the water purveyors that we work with are just using this for everyone. Why give 2? We just are going to go by that one. So like I said, technically this isn't legally enforced, but a lot of people are taking this very, very seriously. For the recreational water bodies, here are the freshwater level criteria. So microcystins greater than 4, cylindrospermopsin greater than 8. So not to be exceeded per day, but then they do have, how many, in terms of a recreational criteria, how many times can you exceed that? And this approach is similar to fecal coliform or E. coli that's being used on a beach area. So it tends to be, in this neck of the woods, it's between Labor Day, it's between Memorial Day and Labor Day, where people are really concerned about people going into water from a beach area. So a lot of the recreational water bodies, state park, are wrapping this in very similar to the way they deal with E. coli, contamination for beach use. But we're really focusing on the drinking water supplies. Again, here are the criteria, and like I said, that the drinking water Water purveyors are really just focusing on these lower ones for everyone. They're not going to tell someone your baby can't drink the water, but go ahead, you can have a glass. They're just going to focus really on that lower criteria. And so to deal with these blooms, I just want to see how I'm doing for time. Yep, okay. So we developed a program called the PARE program, which focuses on predict, analyze, react, and educate. It's a way for water purveyors or a state park to really get a handle on their harmful algae blooms, specifically, the cyanobacteria. Predict is essentially one of two things. Look at the existing data you have. You could have existing data on phosphorus, on temperature and DO profiles, remote sensing technologies. You may have some in situ data, again on dissolved oxygen temperature, looking at the weather patterns and when you see a bloom occur. See if with the existing data that's either available or you have, can you see a pattern in terms of when these blue-green algae blooms hit? The next phase is to analyze. Okay, if you recognize you have some data, but you have data gaps, or you really need to focus on the collection of some water quality data, what type of data do you need? And again, it really depends on the system. For most drinking water purveyors, they're really focusing on phytoplankton ID and quantification, measuring water clarity with a Secchi depth, measuring chlorophyll A, which is the amount of it measures the amount of algal biomass. Measuring taste and odor compounds can certainly at least give you an idea when the blue-greens are showing up, but it doesn't necessarily mean you have cyanotoxins there. Focusing more on the cyanobacterial cell counts, because you'll see certain thresholds result in certain levels of probability of whether or not you're going to have cyanotoxins. And I'll touch on that in just a minute. But what I'll focus on a little bit here is actually measuring the microcystins and cylindrospermopsin. There's all kinds of ways to measure it. There are quick and cheap ways, but again, you have to sort of take these with a grain of salt. A lot of times, very frequently, we'll use this test strip criteria. We link it to our collecting of water quality data. Anytime you're collecting samples for cyanotoxins, and this happens a lot, we'll get a sample sent to us because they want us to do an analysis, and it's in a plastic container, and plastic containers don't work. The cyanotoxins will stick onto the container. You need glass containers if you're going to sample for any sort of cyanotoxin, and then you have to either refrigerate or freeze until analysis. The one that we frequently use, again, for the field are these little test strips. They're actually sort of an immunoassay. In a way, they're very similar to like a pregnancy test in that you're adding a number of compounds in, you're adding the water, and then you're seeing whether you can actually visually see whether there's a compound there. We tend to use this as a yes or no. It will have a limit of detection, and usually, depending on the kit you purchase, it will have a range, but usually the limit of detection is at 0.3, which is important because that's where the health criteria is for microcystins. It's 0.3. So this is good as a preliminary screening. That's how we use this. If you get a hit, that's when you may want to send a sample to a lab to do actual, some ELISA or high pressure liquid chromatography to definitively measure that concentration. But we use this for the most times to see, okay, we got a bloom going on, do we have cyanotoxins? A step above that is for Abraxas, where we buy these test kits, as opposed to visually looking at the test strip, you can actually get a meter that will measure it, and it helps to avoid those false positives, because we have occasionally gotten false positives where we go out and retest. So we've been testing with the test strips for about 3 years, and of all the testing we've done, about 3% of the time, we've gotten false positives. So again, very important. This would help, having a meter like this would help to eliminate that issue. And then of course you get into the high-tech lab testing. More costly. Unfortunately, a lot of times this will take days to sometimes even weeks to get the results. And if you have a drinking water facility or if you have a beach and it's 4th of July, you want to know very quickly. So that's why we use the test strips, to get a handle on it. If need be, then we'll send it to a lab. But very frequently, even just based on the appearance, I know New Jersey DEP has temporarily closed beaches because they're concerned about cyanotoxins. Their logic is they'd rather be safe than sorry. If they have to wait for a few days or weeks, they're going to do that. But I know DEC, their state labs, do a lot of their cyanotoxin analysis. Greg Boyer does a lot of, almost all of the state's work. And again, you can imagine he's a busy guy, so you're not necessarily going to get that data right away. Or again, using the test strip is more of, and it's something that anyone can be easily trained to do. It's nothing that, you need you purchase the kit and you can do the analysis. You just have to know that it has its limitations. Very frequently, particularly our drinking water clients, they'll use levels of cell counts to determine whether or not to actually measure for cyanotoxins. So we have a number of clients, they send us their samples and we do the analysis. And if we hit a certain threshold, and usually it's this one, if we see that the cell count for potentially toxic cyanobacteria hits 15,000 cells per mil or greater, that's when we say, okay, let's start testing. And like I said, we've been doing this for the last 3 years, and that really seems to be a good threshold. This threshold of 2,000 to 5,000 cells, based on our experience, that has never resulted in measurable amounts of cyanotoxins. And a lot of this just has to do with the sheer number of cells in the water. So you can imagine, either in a river or a reservoir system, very frequently where the intakes are is where the wind is sort of pushing up along the shoreline. Well, it's concentrating all the cells. So if you're sampling the lake, reservoir, or river in the middle, you may say, oh, there's no cyanotoxins here. But just the fact that all the cells are being concentrated along the shoreline, where that intake may be, that can be an accumulation where you have enough of the cyanotoxins building up. And again, all those cells scrunched up, high UV light, it's going to start to break down, it's going to start to die off, it's going to start to sink into the bottom. So that's where you're going to have a lot of those impacts. I did see, and I meant to reach out to Ann and Yvonne, and I did not, but they had a really cool paper with the Philadelphia Water Department where they're working with flow cam technology to actually get an idea of algal monitoring in the Schuylkill River. And again, I was at the Pennsylvania Lake Management Society meeting a couple days ago, and I cannot remember her name, but she was from Sea Grant. She was using very similar technology of this flow cytology type of work where you're actually putting information into a computer program, and you're running these flow cams where based on the database you have, it will determine what the shape of the algal cell is, and then it will tell you what genus it is. And the advantage to that is it's very quick. So they can put it in and they can tell you, oh, we got Microcystis in the water column. Right there, they can either decide, hey, we better test for microcystins, or maybe we should close the beach down or something like this. So this is almost, as opposed to me getting the sample, processing it, looking at it underneath the microscope, which takes, I can get a turnaround time of a few days. But even that, like 2 days, can be something that's really concerned. So water purveyors are looking into these flow cam technologies where they can literally put the sample in, get an idea of what type of algae is out there so that they can do some real-time decision making. So to react, so what we need to understand in terms of reacting. Well, there's all sorts of issues in terms of the biology I'm not going to get into. A lot of it has to do with nutrient availability. So toward the end of the talk, I'll talk a little more about watershed management and planning and why that's so important. In terms of treating the water, whether it's the raw water or the processed water, very frequently alum or alum surrogates are used to strip the water of the particulate material, and anytime you can do that, if you have those algal cells and strip them out, you can remove that potential source of cyanotoxins, such as microcystins. And I know a lot of the water purveyors that we deal with, if they get a bloom and they get cyanotoxin hits, typically they can treat it so it's coming out clean. We, so far haven't had any issues where it's literally coming out finished, where we're having measurable amounts of cyanotoxins. But a lot of them are focusing now on raw water because they don't want to get in that situation. I've had a couple people come to me and say, if we ever have cyanotoxins coming out in our finished water, I will lose my job. So we want to make sure we're not having that. So again, there's the raw water aspect, and then there's the treatment facility aspect. So the reacting, in terms of watershed management programs, that's where we're trying to reduce the bloom frequency and intensity. And then you have these nutrient sequesters, which I talked about, such as polyaluminum chloride and iron. There's other issues, like I mentioned, in terms of aeration. Even in a riverine system, if there's where that intake is, if you know during August and September it slows down and you could actually have thermal stratification in that section of the river, having some sort of de-strat to keeping it well mixed can certainly minimize the impact. Then you have ozone and oxidizers and even turbidity curtains, putting them up certain times of the year to avoid all those algal cells to accumulate along where that intake is. So those are some of the reacting issues. I know a lot more in reservoirs and lakes, the use of copper sulfate. We really try to minimize the use of copper sulfate because when you hit an algal cell with copper sulfate, the cell bursts and you're putting all the nutrients, all the taste and odor compounds, all the cyanotoxin compounds into the water column. So really, depending on when you're hitting a bloom with copper sulfate, you may actually make conditions worse and not improve upon it. And usually, and again, I know this is a lot easier to say, especially when I talk to our operators in our company. Usually what you want to do, if you're going to actually treat, you want to treat as the bloom is starting. You don't want to hit it when it's in full bloom. And unfortunately, people want to treat right when the water looks disastrous. It's better to treat early on. You use less product, and you'll have more of an effective effect that you're not putting so much of these compounds into the water. So anytime you can do that. Unfortunately, to do this, you needed some decent amount of data. When do your blooms usually come in? Do you have a good idea? Do they usually start early July? Do they start, early August? And then finally, education and outreach. And this is something when you look at the EPA documents with cyanotoxins that they're really focusing on to make sure that you educate whether it's people recreating at a lake or if it's your customers who you supply water to, that you let them know about cyanotoxins and what you're doing to avoid those blooms and what they can do to avoid those blooms. So, shoreline buffering, fertilizer management in the more suburban or urban rural areas, septic management, waterfowl control, trying to manage that on a watershed basis. So implementing PARE, like I said, typically we, depending on the data you have, you may want to start early in the season. I know we have certain water purveyor clients that we absolutely know when we hit 4th of July, after 4th of July is when the blooms are going to hit. So we know typically, April, May, June, we'll go out maybe once a month to take a look. We'll have diatoms, greens, but we know once we hit 4th of July, that's when we have to increase our intensity of monitoring. So having an idea of when and where to sample. And you may start out with the test strips, just to get a sense of what's out there. And if you have a bloom, documenting it, even just a photo of it when it happened really goes a long way, all the way to actual collection of samples for analysis. But putting it into action, making sure that once there is an issue that it's responded to. So whether it's at raw water at a beach or raw water at an intake, that everyone is cognizant of it. Again, talking with our drinking water clients, what they'll do very frequently is they'll sample out in the water, raw water, and if they get a hit, that's when they start sampling immediately in the finished water. And this is just showing you what the test strip looks like. So the control line, the test line, when the test line gets a little lighter, that's when you see a higher concentration. This is just showing, this is from 3 years ago from a New York water supply. This was just the surface, and this is the intake. Here they actually had in the raw water elevated concentrations of microcystin. Now we did after that we did the final test strip and they had none coming out. But that's something that is an issue of concern, and that's why we use this testing to really get a handle on whether or not these compounds are out there. This is just showing you can actually measure total and dissolved. And here, if it was total, and you can see that the concentrations were really elevated. And again, this is drinking water, so here's 0.5. You can see in the raw water, we were well above that 0.3. And a matter of fact, in the raw water, the dissolved went up to 0.5 at one point, and then it went back down. So this is telling me most of the microcystins are in particulate form once they went up dissolved. But this is something that we were concerned about. And while we were doing this, they were sampling in the final water, and it did not hit the final water. So they, again, they're very cognizant of what's going out in the raw water supply. This is just showing some sampling we did. We did a real intensive sampling for a New York reservoir, and this is showing all mid-lake. We took the mean concentration. Again, remember 0.3. So you can see we're consistently below 0.3. We did it at the intake too, consistently below 0.3 at the intake. But when we went out into the watershed, this East Branch Cove, which is an inlet coming into the lake, we actually had elevated microcystin in that branch, in that part of the watershed. So what that has spurred the water company to do is, and we compared it to some other tributaries going in, we've actually focused more on watershed management approaches to reduce the nutrients coming in that sub-watershed because of this. So again, this can be used not only to determine what the quality of the water is going through the intake system, but where are the source of the problems. And this wasn't directly related to the amount of algae biomass either. This was, the highest amount of algae biomass was on the northern end of the lake. It wasn't at the East Branch Cove. But this East Branch Cove was generating some pretty nasty cyanotoxins. So again, I just wanted to show this EPA World Health guidance that you can find that if you're not measuring microcystins or chlorophyll A, if you're just looking at the number of cyanobacteria cells, from a recreational standpoint, you can use this to gauge the probability of health effects. Again, I know a lot of our recreational water body clients will use that criteria. I'm going to wrap up just talking a little bit about some watershed management techniques. I'll be talking about some ultra-urban BMPs, focusing mostly on that. So one of the big problems in very urban areas is the combined sewer overflows. So it's when the sewer system is semi-connected to the stormwater system. And so what happens is it's generating huge amounts of pollution during storm events, which exceeds the sewage treatment capacity. Really to give you a good sense of this, so this is for Philadelphia, that about 60% of Philadelphia systems are combined sewers, about 40% are separate. So you can see during a dry condition, there's no problem going on. The sewage, the sewage is being treated in the wastewater treatment plant, no problem going into the river. But when there's a storm event, in wet weathers, in a separate system, the storm event, the stormwater system will treat or convey that water into the receiving waterway. But the problem with a combined sewer system, sewer overflow, is both the sewer and the stormwater, if it hits a large enough capacity, it will exceed what that wastewater treatment plant can do. And so a portion of that untreated water can go into the receiving river. So that's, for a very urban situation, that is something that people are very concerned about. So, sewer separation is obviously the best way to go, but as you can see, very expensive. So for small neighborhoods, in one study I looked at in DC, it was estimated in 2011 it cost $11 million to do this sort of infrastructure modification. Also, anything you can do to sort of manage that water, store and treat and maybe disinfect. But again, now we're talking sort of larger, more expensive ways of dealing with it. Some lower cost ways of dealing with it is, maybe having some moderate treatment of the combined sewer overflow. Maybe you can't get it so that it's complete drinking water quality, but at least maybe you could get some of the really nasty solids or some of the particulate, if there's some particulates or trash, remove that. Also managing the source of the stormwater, and I'll touch on that in a bit, and then also gray versus green infrastructure. And again, that keynote speech about forested land, even in an urban environment, anywhere you can put vegetation, anywhere, that vegetation helps to suck up the water, put it back up into the atmosphere, and it helps to absorb the nutrients. So anytime you can make anything green, that's good. So again, for ultra-urban BMPs, there are a variety of practices. Again, I'm not going to get into it. I actually teach a class on watershed management at Del Valle University, and this will be one of the talks. One of my presentations is all on urban BMPs. But essentially, what I do want to convey to you is why vegetation is so important is when you have a natural setting, when all that water, rainwater, hits a forested area, the majority of that water is either going into the ground or it's going back up as evapotranspiration. So that very little is running off, only 10%. As you build and you have more impervious surfaces, it doesn't allow the water to go into the ground, and there's less evaporation because you don't have the trees to help pump that water into the atmosphere. So that means we went from 10% to 55% runoff. So anything you can do to reduce that runoff. Now, agricultural runoff, we're familiar with some sort of unmanaged agricultural system. You have a lot of suspended solids, a lot of nutrients. But again, in an urban system, we see this as well. We see, a lot of trash, a lot of debris, solids, nutrients, all washing into these systems. And I'll just conclude by, some ideas that could be done to help mitigate this in very urban environments. Porous pavement and rain gardens, bless you. Anything you can do to put vegetation in, to put water up into the atmosphere and to absorb those nutrients will really go a long way. Rooftop gardens can be very effective at eliminating that water load. So in conclusion, blue-green algae, they certainly can bloom in riverine systems, and certainly the Schuylkill River does have the potential to experience such blooms with elevated water temperatures, low flow, and elevated nutrient concentrations, particularly phosphorus. The blue-greens do have the potential to generate cyanotoxins, which have a negative impact on humans, livestock, and pets. We developed a PARE program, which can help to guide water purveyors in proactively dealing with these blooms. However, in order to minimize the exposure to cyanotoxins, both water purveyors and watershed stakeholders really need to minimize excessive non-point source pollution. Again, it's one of the things we're all talking about here, why the watershed's so important. So if you can reduce the load going into those receiving waterways, you'll help to minimize or avoid these cyanotoxin producing algae blooms. So thank you very much. I appreciate your attention. Thank you.
We have about 5 minutes for questions, and if anybody needs to sign in for CEU credits, I'll just be over here with it, and then you can come and see me and do that. So we do have a few minutes for questions, if you'd like.
Yeah.
It might be obvious, but these are natural occurring in all water bodies. They're not
Yes.
like invasives where they can be transported. We don't need to worry about
Yeah, I mean,
They're there.
algae are cosmopolitan. They're very global. Once in a while, there's a rare, like green algae that's found in a, like there was this one beautiful filamentous blue-green algae called Staria found in an African rift lake. It was a beautiful, but it's only found in that lake. Almost all these genera are found globally. They're found all over. And so far, there's no definitive pattern of seeing more cyanotoxins in one continent versus the other. It's usually more blue-green algae, the higher the incidence of cyanotoxins, and it's usually related to nutrients and weather.
Can the duckweed and the cyanotoxins coexist?
They can, but usually what we see, at least in smaller systems, if duckweed's taken over, it's difficult for the blue-greens to really take off, because now I've seen small ponds where duckweed completely takes over. And what happens with that is now there's no oxygen transfer, and you have a fish kill because they're not letting anything grow. So usually there's some level of competition. So they were very happy that it was duckweed and not blue-green algae.
I have two questions that are kind of related. Do you find more of a harmful effect from the cyanobacteria when the algae is living versus when it's killed off? And then also, do you have a eutrophication effect when it's killed off, and that it takes up that oxygen like you see in the bay and whatnot?
Mm-hmm. Yeah. I haven't seen anything that definitively says that health impacts are directly related to a healthy or a dying bloom. I do know that I've seen that when a bloom dies, we'll see like that dissolved cyanobacteria. You can get a bump, or you can get a bump of nutrients, and then you'll get a secondary bloom. So I do know that a dying bloom, if it happens early enough in the season that you have plenty of warm, hot conditions, a secondary bloom can occur, which can have cyanotoxins. So it can be directly related. And yes, very similar to marine settings, as that bloom dies off, it's eating up oxygen. So you could potentially, depending on the size of the water body, how low the flow is, and the magnitude of the bloom, it could result in like a fish kill or something negative.
Right. And then greater health issues during the kill, is that what you do?
I haven't seen any studies that said that definitively. I would say that the fact that as those cells are lysing and as you have more dissolved cyanotoxins, that's more of a concern because the dissolved compounds are more difficult. Once it goes into the treatment system, that dissolved cyanotoxin is very difficult to remove. If it's still bound in the algal cells, then like the water purveyors can like settle it, can strip it out of the water column, and so you're getting rid of them. Once it's dissolved, they're a lot more concerned. So that's why in that instance we were measuring both total and dissolved, because they wanted to know.
Fred, tell us something about the regulatory context. A couple of questions that come to mind. One, you mentioned that EPA has some ongoing investigations. Are those still ongoing, or have they been shut down by the current administration? And then a related question is, who is regulating this quality of water in the public drinking water supplies at the states, or who's doing it?
Yep. Okay, so for, I can tell you, as far as I know, the unregulated monitoring program is still going on. I know a number of our clients have received notification that they have to start monitoring. You know, it's going to be over the next 2 years. And the reason why they do that is, you know, EPA wants to collect enough data so that they can then put forward an actual regulation. So right now, everything that's available, whether it's recreational or whether it's potable, these are draft. Like I said, the drinking water companies are taking this very seriously. So what I can tell you is that they'll hire us to do the monitoring. We provide them with results, and then we tell them on how they should deal with particular situations, you know, using that PARE program. In terms of a federal standpoint, as far as I know, there is a lot of monitoring going on for public waterways through EPA. I don't know how that's going to change over the next year or so. The last AWWA meeting I went to, which was for New Jersey, they were saying it's full steam ahead. That was fall of last year. And I'm actually giving like a whole class on this, like a half-a-day class on cyanotoxins and raw water supplies for Pennsylvania AWWA. And it's all on raw water, and that's what they want to hear. And talking to them, as far as they know, they have to comply with this monitoring coming up.
Is regulation going on by the state departments of environmental protection or the health departments, or
well, right now it's like state. So New York actually has some criteria on the books, and so they will shut a beach down. They will tell a water purveyor you have an issue, because like I said, you know, samples are sent to the state. The state sent these criteria. I think Oregon also has its own criteria set up. Pennsylvania currently does not, but talking with PA right now, they're recommending the EPA criteria. And like I said, our clients they're taking it very seriously. It's funny because we've been talking about this for years and years. And I would get this response like, well, if I'm not told I need to do it, then it's not a problem. As soon as these came out, even though they're draft, all of a sudden the floodgates opened and all the water purveyors were like, I want to know about this, I want to deal with it.
Does anyone know why these toxins are manufactured, why the algae produce them?
That's a really good question. The only thing we know is when that bloom feels stressed, and that's it. I mean, as many papers as I've seen that says when you get high temperatures, that will trigger a bloom. I've seen it in as many papers say when temperatures decline, you get a bloom. So there's no overarching predictive model. The only thing we can say is if for some reason that bloom feels stressed, it starts to trigger the bloom. And that doesn't even necessarily mean that's going to happen all the time. So that's something that there's a lot of money, a lot of time, and a lot of effort being placed to try to find some definitive correlations.
Do you think nutrient limitation might trigger it?
Yeah, I mean, beyond a shadow of a doubt, the one thing we do know is if you increase nutrients, particularly phosphorus, you'll get more algae, and more phosphorus tends to mean more blue-green algae. So that's pretty definitive. The other thing too is those three conditions. My mantra, you know, hot weather conditions, still water conditions, high phosphorus concentrations will almost always in freshwater systems result in blue-green algae blooms. Yes.
You mentioned that the toxins stick to plastic.
Mm-hmm.
How effective is that? Would that be a viable filtration option?
No, that's actually an interesting point. I know the company that makes the products are the ones who discovered the whole plastic issue. But no, that's actually a pretty intriguing way of maybe removing them out of the water column. You may actually have a patent there. I've never heard of that, but that's a pretty cool idea. Yeah. Yeah, plastics. Yeah. I'm sorry, what?
Did they happen to notice the difference between different types of plastics or not there yet?
The plastics used for collecting water samples, that's the only thing I know. And so just like about 4 years ago, they just came out with this big mantra, like from now on, everything has to be in glass. We've seen, I mean, they had arguments with some people in Toledo about the collection because they kept collecting with plastic, and they're like, no, you can't collect with plastic, you have to use glass. And they had to do this real definitive study to show the data, and they were like, okay, we know we have to collect with glass now. Right? All right.
Yeah.
Thank you, appreciate it.